started.
Lab 5, Part 4, Respiration, by Bobby Puishys and Sam Kesseli.
To introduce the lab, you must first understand that the human body is dependent on the intake of oxygen and subsequent release of carbon dioxide in respiration. The lungs are responsible for carrying out the process of gas exchange. The process of cellular respiration binds carbon from glucose to oxygen, forming carbon dioxide, which is released as a waste product of the electron transport system. The purpose of this week's lab is to analyze how factors such as exercise affect this process, and to compare the concentrations of carbon dioxide in room air to that of exhaled air. In addition, the vital capacity of the two subjects was determined and compared with the averages for the class.
The objectives of the lab are quite simple. In Part A, the objective was to compare the CO2 concentration in room air with that of exhaled air. In Part B, the objective was to observe the effect of exercise on carbon dioxide production. And in Part C, the objective was to determine the vital capacity of the two subjects and how it compares to the other members of the class. So, a little background on the respiratory system. Its main function is to bring oxygen into the body and remove CO2 from the blood. It relies on a vast system of capillaries which run through the alveoli of the lungs. The alveoli basically facilitate the gas exchange to and from the blood. They have large surface area and are pretty foamy, which allows the maximum exchange. So during inspiration, oxygen is obtained, and during exhalation, CO2 is released. In this lab, phenolphthalein was used as the indicator of acidity, and it basically indirectly indicates CO2 concentration. Additionally, in the first part, lime water was used as a simple indicator for CO2 level.
So Figure 1 shows the exchange of gases between the blood and the alveoli. Cellular respiration requires oxygen to accept electrons in the electron transport system within the mitochondria to produce ATP in cells. Carbon dioxide is released as a byproduct of this process. Exchange of these gases is thus critical in obtaining cellular energy. The alveoli in the lungs enable this exchange in the blood. The large surface area of the alveoli enable maximum solvation of oxygen into the bloodstream and diffusion of CO2 out to be exhaled.
So, before the experiment, a number of hypotheses were made. For Part A, it was hypothesized that exhaled air will have a higher CO2 concentration than room air. So, ultimately, the lime water solution will turn foggy. For Part B, it was hypothesized that the CO2 levels in the exhaled air will be higher after exercise. And for Part C, it was hypothesized that the vital capacity will be dependent on gender as well as size.
A summary of the results we found. Part A, we found that exhaled air contained higher levels of carbon dioxide when compared qualitatively to room air. Part B showed that the air exhaled after exercise had higher CO2 levels, again qualitatively, than the air exhaled before exercise. Both exhaled air tests revealed that they had higher CO2 concentrations than the room air. Part C, lastly, we found that vital capacities are nearly double those of the smaller members of the class for tall males. All three results appear to support our initial hypotheses.
So the lab was performed in accordance with the procedure outlined in Part 4 of BB2903 Lab 5, Fetal Pig Dissection Part 3, Circulatory, Respiratory, and Nervous Systems, without any modifications.
For the first experiment, we wanted to compare the CO2 content of exhaled air versus room air. Lime water was used as an indicator, and exhaled air was first blown through a straw into the lime water. Table 1 records the results of this experiment. The exhaled air produced a cloudy solution within the beaker, while when the room air was bubbled through, no change in solution opacity was observed. This experiment qualitatively tested the concentrations of CO2 in the two kinds of air.
So, in Part A, the hypothesis was confirmed. The lime water turned cloudy when air was exhaled into it, but not when room air was forced into it. This occurred because of the formation of calcium carbonate. Calcium carbonate forms in lime water when it's reacted with CO2. When the reaction occurred, it indicated to us that there was a high CO2 concentration in the lime water.
Experiment B tested the carbon dioxide concentration in exhaled air before and after exercise, with room air as a control. The results for this experiment can be seen in Table 2. Phenolphthalein was used as the indicator in this test. A phenolphthalein solution becomes clear when it is acidic. This acidic concentration increases during carbonic acid formation when exposed to carbon dioxide. The number of seconds in this table indicates the amount of time air was bubbled through the solution before it became clear. Drops was the number of drops of ammonia needed to return the solution back to pink. And finally, the estimated carbon dioxide content was calculated with a normalizing equation to see the relative qualitative concentrations of CO2 for each test.
So in Part B, a clear difference was observed in the times and the number of drops it took for exhaled air, as well as exhaled air after exercise and the room air. The volume of ammonia required to bring the solution back to pink was more for the exercise solution, which directly confirms the hypothesis. So it was shown, basically, that the exercise air did have a larger amount of CO2. So a simple ANOVA was performed to confirm that the results weren't due to chance, and the p-value turned out to be 1.76 times 10 to the negative 12, meaning there's almost a 0% chance that this could be due to chance. The ANOVA also gives an average for each of the values. You can see the average for room air is 6.6 drops. This is for drops to get it back to pink. And then the exhaled air was 104 and the exercise air was 162. So you can see there's a large difference. This is also shown again here by the graph. You can see that room air is a very low amount of drops, exhaled air is in the middle, and exercise air is a much higher amount of drops.
Part C experimented the vital capacity of two subjects with a device called a spirometer. When a spirometer is exhaled into, the air is collected in a bucket and a readable measurement is given as the output. We performed 3 trials on the spirometer, each measuring vital capacities between 5.5 and 5.7 liters. Both subjects were 20-year-old males, approximately 75 inches tall. Figure 7 shows exhalation into a spirometer. According to medical physiology, a clinical text, the average vital capacity for people is 4.8 liters. The average vital capacity for the students in the classroom was 3 liters. Both subjects had vital capacities higher than the classroom and textbook averages. It's important to note that both of these subjects were consequently tall males, which account for these high values. These high values confirm the hypothesis that the vital capacity is dependent on size and gender.
So from the lab, a couple conclusions can be made. We concluded that cellular respiration is a vital process, and it requires oxygen as well as glucose, and ultimately the byproduct is CO2. Exhaled air has a high concentration of CO2 since it is, once again, a biological waste product. Exercise affects the level of CO2 output since it increases the rate at which sugars are metabolized. This increased output is measurable, and as we did in this lab, it can be measured using lime water or phenolphthalein. And vital capacity appears that it is dependent on height, weight, and gender. These are our references. Thanks for watching. I'm going to stop this.